My name is Guo-Ping Shi from the Cardiovascular Medicine, Brigham Women's Hospital, Harvard Medical School in Boston here. And I came to present our work about regulated T cells in abdominal aortic aneurysms. And we initially started with humans. We found that we screened about 50,000 patients, and among them about 500 patients with aneurysms. And we used 200 age and gender matched controls to find that the regular T cell numbers, contents are significantly reduced in those AAA patients, and correlated significantly but negatively with aneurysm growth rate. So we suggested that those T regular cells may protect aneurysm development in humans. To test the hypothesis, we used NG2-induced aneurysms, and by giving those mice Treg cells and to protect aneurysm development. As a matter of fact, we can do that, and when we give the mice with Treg cells, we are able to reduce the aneurysm development. In terms of mechanism studies, we found that a lot of people believe that the Treg cells produce IL-6, IL-10 as anti-inflammatory cytokines. So what do cytokines do? And we are able to prove using IL-10 knockout mice and the Treg cells from those mice and do cell repopulation tests to show that Treg cells release IL-10 as anti-inflammatory molecules to reduce inflammatory cell accumulation in aortic aneurysm lesions, such as the macrophages, CD4 cells, and the CD8 cells, as one of the mechanisms. The second mechanism we've discovered is that those cytokines from Treg cells controls angiogenesis. Therefore, when we repopulate IL-10 knockout the Treg cells into those NG2-induced aneurysm mice, such cells lost their activities because they do not have such molecules produced. So we use those cell population method and the molecular, angiotensin-2-induced experimental models to prove the role of aneurysms to define the mechanism of such cells, how they, protect those disease in mice. Hopefully this will be true in humans. At least in humans, we found a reduced number of such cells, and we hope that this will give us another potential to define the mechanisms and the potential therapies for human abdominal aortic aneurysms.